So I, don't have to start with the usual introductory slides about insects declining to this audience. So I'm going to do this very quickly. But I think one of the main important things, which I'm excited to hear about in the next webinar, are all of the different, 852 cuts that insects are having to survive. But I want to argue that insect long-term data sets are rare. And because they're rare, museum data and community science data is actually really useful for understanding patterns of biodiversity, particularly for insects. And, now in GBIF, there's like in the Global Biodiversity Information Facility, over 2.5 billion occurrences. Most of them are for birds, but many of these ones are for insects. And the great thing about this data is that it's occurrence data. It includes many different sources, such as digitized museum records, surveys, and community science data, such as iNaturalist data, and it's rich with metadata, such as like species names, location, and sampling date. And the main problem is that it's presence-only data and that we don't have a measure of effort. And so, for example, if we look at this data naively, so this is the number of records for bees in the United States, you can see that the number of records has increased tremendously. And if we analyze this data naively, we would infer that, bees are increasing in abundance. And we have the same problems across space where, this is the exact same data plotted across North America. And you can see that, if we were to analyze this data naively, we would say that Los Angeles and New York City are biodiversity hotspots for bees. And so, oh, come on, I can't advance slides now. Oh, there we go, here. So I would argue that we need a general approach to analyze this unstructured data, and I think occupancy models are that tool. And so I know that you came here today to listen about pesticides, but I want to first introduce you to occupancy models and how I've been using them to model unstructured data, because then it's going to be very useful to understand the study that I recently published on looking at the impact of pesticide use for bees in the United States. Okay, so why occupancy models? So occupancy models are really useful because they allow us to model sampling effort that can vary across space and time. So the 101 on what an occupancy model is. So imagine here that we set up multiple different sites and we visit those sites at different time intervals. And so because our ability to detect species is not great, when we have these structured surveys, our detection data will look something like this, where a lot of the times we're going to miss species even when we know they're there. So for example, here in my site number 2 and site number 3, I know that the species is there because we've observed it in different time points, but we have missed detections. In site number 4 is a little bit harder because it could be that the species is truly absent from that site. And because our ability to detect species is quite high in this scenario, we're quite confident to say that the species is absent. But when our ability to detect species is poor, disentangling this missing absences from missed detections or, from non-detections is a little bit more challenging. And that's exactly what occupancy models help us do. And so essentially, occupancy models are a set of 2 logistic regressions where we're modeling the true presence or absence of a species at a site, which we modeled, we estimate the true, the occupancy probability of that species using a Bernoulli distribution. But our actual observations are actually dependent on both the true latent state, whether the species is there or not, and the probability of detection. Now, the main problem that we have when we have presence-only data is that somehow we have to infer absences, right? In the naive approach, my we might be able to take all of the data from GBIF and bin it across space or across time. But filling those site and time interval combinations with zeros might be misleading. Mostly because some of these sites and time intervals may not have been visited, or they're visited and not databased, or they're indeed visited and we didn't find the species. And so hopefully this graphs convince you that we can actually use information from other species to infer visitation. And so I've done some extensive simulation studies looking at how robust are these models when we infer absences based on others on the detection of other species, and when some of that visitation is also targeted, when we know that we have, say, iNaturalist data combined. And for the most part, we find that these models are quite robust when we infer absences using other species detections. Okay. And also to try to convince you some more, we've simulated scenarios where we know that not all visitation events targeted all the species, but sometimes target a single species. And when at least 50% of the sampling events come from multiple, like from visitations that look for multiple species, you can still infer absences based on other species presences. Okay, so this is like the fastest overview I've ever done on occupancy models, but that's because I want to get to the meat, the meat of the problem. So essentially what we're doing here is that we're using multi-species occupancy models because they're more robust. They allow us to estimate detection for multiple species across a site. So they allow us to share information across multiple species, and we're using the absences or the presences from other species to infer visitation for any species of interest. Okay, so with these tools in mind, I was really interested in understanding the role of pesticide use, specifically in shaping the distributions of North American bees. And we know that there's many different drivers that are affecting the occupancy of bees, but many of these drivers are associated with agricultural intensification. And so with agricultural intensification, we've seen the reduction of floral and nesting resources by the conversion of different types of lands, reducing floral resources. But more importantly, with agricultural intensification, we've seen different types of shifts of pesticide use, which exposes bees in the US. And in particular, there's two classes of pesticides that have become very prevalent and important in the story. These are neonicotinoids and pyrethroids. And so in this map, I'm showing you the amount of pesticide use combined between all compounds of neonicotinoids between 1995 and 2013 using the USGS pesticide use project data. And you can see that since they were developed in the late 1980s, neonicotinoids were adopted really rapidly across most of the United States. And just so you know, these are log scaled. And so the amount of these compounds used across the country is pretty high. And the other problem is that neonicotinoids have many different exposure routes for bees. They're sprayed, they are applied to soil drenches, but they're also applied to seeds. And when they're applied to seeds, they become systemic, meaning that many different parts of the plant tissue have these compounds in the exposed bees. And the other reason why I wanted to focus on pyrethroids is because they're the second most commonly used type of insecticide in the world. And previous studies have looked at the composition of insecticides since the mid-1990s in the US, and they found that most of the risk for insects, particularly bees, comes from the huge uptick of neonicotinoids and continued use on pyrethroids, and less so by the use of organophosphates or carbamates. And so because of this, I focus mostly on these two types of insecticides. The other thing is that crops that rely or that benefit from animal pollination sometimes use managed pollinators like honeybees to increase pollination. Now, one of the problems is that honeybees have been shown to compete or transmit disease to native bees, but also crops that use honeybees may have different pesticide use patterns. And so actually disentangling all of these threads is really challenging. And one of the main problems that we have is we have a lot of data gaps, even when we use all occurrence data that we have. The other thing is we have a lot of evidence of experiments looking at the effect of pesticides on bees, but less so understanding how that is shaping kind of distributional trends of these species. And so even in like the United States, where we have millions of records of bees, we still have a lot of sampling gaps. And so with a study that we did with Paige Cheshire et al., we were looking at the completeness of records across the board. And depending on the spatial resolution, the completeness of these records goes anywhere between 5% to 40%. And you can see that there's some areas of the country that have not been sampled very well. And we have a lot of evidence linking pesticide use to linking the effect of pesticide use to bee declines. Many of these are coming from experimental studies. But it's important to, in addition, looking at effects of mortality and reproduction, to look at the combined effects on distributions. And so what I wanted to ask is whether pesticide use could be drivers of continental white decline. And so to do this, I used essentially all or as much of the available data on occurrences for bees that we have. You can see that the distribution of occurrences is not very equally well distributed across all the species. So for example, for aphid bees, we have a lot more records than the expected based on the number of species, whereas for megachilids, we have a lot fewer records than we would expect based on how many species we've got. Now, I couldn't model every single species because there's a lot of species that we haven't seen since 1995. But in this analysis, I was able to include about 200,000 presence, like years by site by species presences for about 1,079 species of bees during the period of 1995 to 2015. And you can see the different, the number of species that I modeled from 6 different families of bees. And so what I did is I used occupancy models, and the ones that I introduced at the beginning, the multi-species occupancy models, and causal analysis to evaluate the role of pesticide and agricultural cover per se. And specifically structural causal analysis. And so because we know that agriculture is not distributed equally across the country, I included things like to what extent climate and topography affect the distribution of climate and floral resources. But also all agriculture is not, it's not equal. A field of wheat is very different from an orchard of cherries. And so I used the USDA attractiveness to pollinators or attractiveness to bees metric to differentiate agriculture that attracts or requires pollinators. And so you can see here, combining this attractiveness with the USDA crop data layer, the distribution of animal pollinated agriculture across the United States. For pesticides, I relied on the pesticide use national synthesis project. And unfortunately, this project stopped recording data on seed-coated neonicotinoids after 2014. And so this analysis was only from 1995 to 2014. And I also tried to obtain data for honeybee colonies, which is surprisingly very challenging to find, particularly before 2016. But here I've relied on the Census of Agriculture, which tells us how many colonies there are per county in certain years. Okay, so on the top left-hand side, I'm showing you the different causal diagrams that I used, both the variables that were included in the analysis and the variables that were omitted or not needed. And so I wanted to know whether pesticides are driving changes in occupancy for bees. And here I'm going to show you family level means. And so what I found was that as you increase pesticide use, both the combined use of pyrethroids and neonicotinoids, it decreases the occupancy probability across space and time for 5 different families of bees, and significantly so for 4 families of bees. But okay, these are kind of like slope coefficients, and you may be asking, like, what does it mean to have like a minus 0.5 effect? So here I translated this negative slope coefficient to a predicted occupancy when you go from a county that has no occupancy, no pesticide use on the left-hand side, to the maximum pesticide use that we have in a data set. Or so either across space or across time. And this results in about a 43% decrease in occupancy probability for aphid bees, 29% for andrenids, 20% for halictids, etc. And so the decline in occupancy probability is quite substantial across this gradient of pesticide use. I also looked at the effect of animal pollinated agriculture per se, and generally the effect is less strong and positive, which is not surprising because this is agriculture filtered by agriculture that attracts pollinators or that benefits from pollination. Now, here, what I'm showing you are general level trends. And so on the top panel, that's the effect of pesticide use. On the bottom panel is the effect of agriculture. The 5 different families of bees are here in the top, and I don't think you can see my cursor, which doesn't matter. And each column is a different genus. And I'm only plotting here genera that had more than 10 species. So for the most part, and above the line, it's a negative effect. Below the line, it's a positive effect because the graph is flipped over. Okay, so the main thing is that across many different genera, we see this negative effect of pesticides. Similarly, we see the slightly positive and less strong effect of animal pollinated agriculture. But I want you to focus on bumblebees. And so bumblebees were really surprising because I found this really strong negative effect of pesticide on bumblebees. Now, when I calculated the actual change in occupancy for bumblebees, it comes to about 54% decline in occupancy probability. And so there may be multiple reasons for this. One, it may be that bumblebees are more sensitive than other bees to pesticides. And there is some evidence for this. We did, with some other colleagues, we did a meta-analysis looking at lethal doses of 4 different types of neonicotinoids across genera for bees. And we can see that Bombus has a lower LD50, meaning that it's more sensitive to acetamiprid and imidacloprid than, say, for example, honeybees. So it could be that bumblebees are more sensitive. And indeed, I was really happy when I saw the study by Nicholson et al. Earlier this year, because they also found, relatively consistent patterns in the sense that they looked at the effect of pesticide risk, the cumulative effect of all different compounds on total production of bumblebees and queen production. And they found also a 52% reduction in total production and a 47% reduction in queen production. And so the estimates in occupancy probability are approximately in the same order of magnitude of what we would expect from experimental studies. But the other thing is that despite the fact that I used records or as many occurrence records as there are available for bees, bumblebees are about 46 species here in North America out of the 3,500 for which we have data. And yet they comprise 25% of all the records. Which means that maybe we have the best ability to detect this signal because of the high number of records. And so from this like really sped up talk, the main takeaways is that I think we do have evidence from this occupancy framework that neonicotinoids and pyrethroids are a driver of changes of distributional effects in bees. Bumblebees could be more sensitive to pesticides, but they could also have the best data to detect this effect. And animal pollinated agriculture had a slight positive impact on these distributions. And with that, I'm happy to stop and take any questions or go up for discussion.